All right, I have uploaded the previous video about cells of the innate immunity internal defense. Innate immunity internal defense. So just to revise again this classification, immunity, two types. One is innate, other one is adaptive. Innate immunity, again, we divide into a external and then internal. Adaptive immunity we can again divide into two, cellular and humoral. Cellular and humoral. Now internal defense, we largely talk about the inflammation. So we have discussed inflammation in the previous video. You can watch these videos, two videos about inflammation. So inflammation involves chemicals and cells. So we discussed about innate immunity external in the first class, and we also discussed innate immunity internal, inflammation, cells. What is left to discuss? Chemicals in the inflammatory response or internal defense, and after that we can talk about the most important part, adaptive immunity. So I will revise several times this classification in order to understand where we are in the lessons. So it is also easy for you if you can remember this flow chart, then you can easily understand when something you're going to read, okay, this is innate immunity, this is external, internal, inflammation, chemicals and cells. Adaptive immunity, cellular and humoral. Since I have discussed about this phagocytic cells, natural killer cells, in terms of inflammatory response, I'm going to discuss today chemicals, and there are many chemicals. We only discuss interferons and complement proteins. So we're going to discuss two main chemicals which are very important in inflammation and innate immune response. So we're going to discuss these two chemicals. Right, first I'm take the interferon. This is usually abbreviated in many textbooks IFN. Interferon. Now, interferons are very important in viral infection. So, interferons are important group of chemicals in viral infection. Okay, let's see how this interferon work in viral infection. First of all, we should little bit revise how virus looks like. Okay. Yes, just to again revise, you can check the previous videos up to this point. I have uploaded 2 videos, and the 2 videos explain innate immunity, internal defense, inflammatory response, phagocytic cells, and natural killer cells. So this part is covered in the previous uploaded video. So you can check this video to learn this part. Now today we are going to learn about chemicals in the innate immunity internal defense. Okay, so first chemical is interferon. Interferon is very important chemical in viral infection. Now it is important for us to always keep in mind how virus looks like and what is the importance of virus. It doesn't matter what is in the syllabus, day by day, the importance of virus coming to learning points, because of many new viruses will come, and we should know better understanding of vaccine and other things, how virus can infect human cells. So many times we have to talk about virus. Increasingly, exams, they're also checking student knowledge about virus. So we have to learn at many places. For example, microbiology is a complete unit. In that unit, we have to learn a lot about virus. But we learned a little bit about virus previously. And here again, we have to learn about virus, because virus is one of the main infectious agents that can cause diseases in human and other animals. Right. If you remember how virus looks like, every virus, they have a similar things. One of the thing is they have a protein coat like this. Just a protein coat, and the protein coat protecting piece of nucleic acid. So inside piece of nucleic acid. This nucleic acid could be RNA or maybe a DNA. They don't have both. They have only RNA. If not, they have DNA. So this is a nucleic acid and this is a protein coat. And in protein coat, sometimes we can find special proteins. If you remember cell membrane, we discussed about glycoproteins. In human eukaryotic and prokaryotic cell membrane, we have learned phospholipid, and the proteins are the main components. And the proteins, again, we divide into different groups. So you can check this small video I posted about the question, and I revised again the structure of the plasma membrane. Here, there are special glycoproteins we can find in the protein coat like this. So they are again part of the virus protein, and usually they are virus glycoprotein, and they are important for the attachment. Hence, these proteins also called virus attachment proteins. They are glycoproteins. So these are called virus attachment protein. This protein is called capsid. The protein coat is called capsid. The name given to protein called is capsid, and this is nucleic acid. It could be DNA or RNA, right? So this is a very basic structure of virus. After that, we can add certain modification for different group of viruses. But basically, all the virus, they share the common things. It has a DNA or RNA covered, protected with the capsid. This capsid has different proteins, including glycoproteins. They act as attachment to the host cell. And sometimes, when you talk about different viruses for just interest, for example, coronavirus-like things or HIV virus-like viruses, they have again lipid membrane from the host cell. So it's this. So they have again lipid membrane from the host cell. This is some of the example of differences we can find in viruses. Okay, now these glycoproteins should be complementary to host cell receptors. That means every virus cannot attach to host cells. So good example is when you talk about viral disease, some viruses infect only human. It doesn't infect our animals like dogs, cats, cows. They're not infecting because they don't have a receptor to the virus attachment protein. Now that is a problem when we talk about viral disease. We call viruses are not non-living things. They are not living. If they are not living, how come all of a sudden we get a virus disease? New viruses, how it comes? So answer is very simple. Virus, if they want to, as they are non-living things, if they want to still survive, most of the viruses we can find in animals. Otherwise, if the virus stay on a hot surface, you can understand if the temperature is 40 around, the protein get denatured, then the virus is not active. So virus need a right place to live. Most of the animals, viruses are there in animals, but they can't infect because they cannot attach to the host cell. So they just stay there without dying or without destroying its structure. But if they develop a special glycoprotein receptor that can bind to the host cell, then that is a problem. So we can talk about more about this in the future lessons. At the moment, we just talk about this interferon. Now this is a host cell. So there is a receptor which can attach to the host cells. Now, if this fit nicely, this is the only requirement. The virus can go into the host cell. So there are two things can happen. One is this nucleic acid can go into the host cell. If not, the whole virus can enter into the host cell. So depending on the virus, the whole virus can go into the host cell, or in certain situation, only the nucleic acid can go into the host cell. So we have learned virus can, we don't use the word reproduced. We don't use reproduce, we use replicate. You can understand why we use replicate, because it's not a living thing. Living thing can reproduce. So we define living thing. Reproduction is one of the important feature. I mean, unit 1, if you recall, reproduction is one of the important parameter we consider for living thing. So we don't use word reproduce, virus can replicate. So we can learn this later, how the virus can replicate in host cell. Now what's going to happen at the end, when it enter into the host cell, so it start to make many virus. So when it make many viruses, you can understand where can they get the resource, where they get the proteins, where they get the nucleic acid, where they get this carbon, hydrogen, oxygen, nitrogen, like all these new things. They're all taken from the host cell. Now when host cell, it's called actually the host cell is hijacked. So host cell is hijacked and they're called parasite because internal parasite, they're going to harm the host cell because they use resources of the host cell. They use the important material of the host cells, and host cell cannot maintain the basic function. The host cell cannot produce its own proteins because virus is aggressive, virus is very fast, so it uses all the resources now. You know, it make a lot of nucleic acid, then how many nucleotide it should use from the host cells. And when it make proteins, amino acid also used. So finally, host cell is running out of material, molecules to maintain the life. Eventually, host cell die. And then new particles, they release when the host cell die. So likewise, they are going to kill many cells in the body. So if the cells are very important, so this host cell is important cell, then we lost the function. Example, why people die in coronavirus, because it destroys lung tissues. So you have learned alveoli. Alveoli maintain the large surface for gas exchange. So gas exchange surface reduced. The person cannot take enough oxygen. When person cannot take enough oxygen, other cells do not get enough oxygen. If other cells do not get enough oxygen, children, you need to electron transport chain and oxidative phosphorylation, ATP production drop. When the ATP production drop, all cells start to die. So that's how a virus can kill a person. Virus going to infect people. So they are very dangerous because of the simple structure. When it's more simple, it's very difficult to make drugs because we can't find any target molecules. That's why there are almost no or one or two or few drugs available for viral infections. In that case, we still appreciate the role of interferon, natural protein synthesized by the body. Now, what's going to happen in this case? This cell has a few things to do now, the host cell. Host cell has few things to do. One thing is just suicide. They call cell suicide. So when cells suicide, it doesn't provide the molecules to virus to replicate, and the virus released to the outside, then phagocytic cells can catch them. Now, the main mechanism of killing infectious agent using these two cells, phagocytic cells, number one, and sometimes natural killer cells. You can, if you watch the videos, natural killer cells are also important. For example, if virus infected this host cell, then the natural killer cell can come and destroy the host cell. Before host cell getting worse, natural killer cell can kill it. Why is that? To release the virus particles. Now, one of the biggest problems we have in virus when they are inside the cells, it is very difficult immune system to attack. They should come out. If they are inside the cells, there's no access to immune system, the cells of the immune system. So keep in mind very strongly, the phagocytic cells are the main type of cells which kill, I use the word kill, the pathogen. Most of the cells, they can't kill the pathogens. Bacteria or virus, they can't kill. But they only support phagocytic cells to kill the cells. So we have learned many places, phagocytosis. So phagocytic cells are the very strong cells. All other cells and chemicals which trying to give enough support to phagocytic cells to kill them. Now in this case, viral infection, they are inside. So only way is get them out, get the virus out of the cells. It happened in two ways. At the end, host cell realize it cannot survive any longer. Then the host cells start to undergoing cell death and the virus particle released. Second method, before it comes to that stage, some of the cells of the immune system attack the host cells and to release virus out. So we can talk about many places this mechanism. And now this cell is trying to do another trick. So it produces a special chemicals. This chemical is interferon. The chemical is interferon. Now interferon can try to protect this cell. Now you can understand, if you take a host cell, there's not millions of viruses coming at the same time to the host cell. So there are certain level of number of viruses attached to the host cell. Let's say at the moment 100, example 100 virus attached to one host cell. Then out of this 100, they can find the receptors and most of them enter into the host cell. So let's say again 100 virus particles enter into the host cell. Now that time, the cell can also do something. Otherwise, cell is not waiting until a virus destroyed the host cell. So cell produce this chemical interferon. Now what is the function of interferon? Interferon function in two ways. Number one, it gives a signal to neighboring cells. For example, this is a neighboring cells which is not infected. This cell is not infected. So this is uninfected cells. So interferon diffuse to uninfected cells. So I write here diffuse to uninfected cell. So just signal to uninfected cells. It gives signal to uninfected cells, like a warning signal. Warning signal is we are under attack of virus. Please prepare for the situation. Now that's a little bit of exaggeration, but to understand what exactly happening, it is the same message. So uninfected cells, interferon serve as a chemical messenger. When interferon is available in uninfected cells can prepare for the situation. At the same time, interferon also try to protect these cells, infected cells. So second one, protect infected cells. So these are the two mechanisms we can expect from interferon. Number one is, I think, one of the important things, because cell is not infected yet. It gives a signal to the uninfected cells, prepare for the situation via the attack of virus. And infected cells still try to protect from dying the cells from virus attack. Okay, now let's see briefly how can it protect host cells, how can it protect infected cells, how can it protect uninfected cells. Okay, it's like this. This is the, take a host cell infected. With virus. So these are the virus particles. And these are the interferon. Okay, one mechanism is interferon as a protein can produce new group of proteins, antimicrobial or antiviral proteins. I think just write virus, antiviral protein. So interferon is responsible to produce antiviral protein. Now, how antiviral proteins help? In several ways. One is stop, inhibit virus replication. So we are not going to look details. What is antimicrobial antivirus protein, and what is the name of this antiviral protein, and how does this antiviral proteins stop virus replication, inhibit replication, or it could inhibit attachment. Inhibit replication, inhibit attachment. So I wrote two methods. So under that, we are not going to discuss in details. It inhibit attachment of virus. Inhibit virus replication. Now, same thing can be done by the neighboring cells. So when interferon diffused to the uninfected cells, it can also inhibit attachment of virus. So it can prevent entry of virus, or already few virus particle entered, it can inhibit replication. Now, you have to consider these are not 100% perfect mechanism. Why is that? You can easily understand people get infected with virus. And these people still produce interferon. So then interferon try to protect. It's a battle between pathogen and the immunity. So certain people get the disease, certain people not get the disease. These mechanisms are there to protect us, but we can't say these mechanisms are 100% perfect. If they all operate, no virus can make us sick. It depends on the person immunity and also depend on the type of virus. So these are early mechanisms the body is trying to do, because innate immunity is the first line of defense. Then it goes to the adaptive immunity, acquired immunity, second line of defense. So each level, it try to completely eliminate, completely kill the pathogens, completely cure the disease at each level. But these levels are not perfect, then it goes to the next level. Okay, now you understand what is the role of interferon. So you have to understand it like this. Interferon are very important in viral infections. When cell is infected with virus, interferons are produced. There are two mechanisms. One is it protects the infected cells. It tries to give signal to the neighboring cells to protect. And how it does, they can produce another group of protein called antiviral protein. Antiviral protein has two functions. It can inhibit attachment of virus. It can inhibit replication of virus. And there is another mechanism, third one. It can support phagocytes. Now that's a important thing I always telling. Phagocytic cells are the main cell. It can kill the pathogen. So far, phagocytic cells are the only cell can kill the pathogens. So interferon can support phagocytic cells. So that is a third method. It can support to remove the virus from the body. So these three methods come into action, and both cell types try to survive. Okay, this is the end of the interferon story. And coming back where we are here. So we are talking about still innate immunity, internal defense. Internal defense, we can place everything into inflammatory response. So we have discussed inflammatory response in the previous uploaded video. So you can watch this video. And I discussed the role of phagocytic cells and natural killer cells. Under chemicals, I discussed about interferons. In the next round, I'm going to discuss another group of proteins called complement proteins. Complement protein. Right now, where can we find this complement protein in the body? So I just write like this: location, plasma. So these proteins are in plasma as inactive proteins. Location in plasma as inactive proteins. So they can activate during inflammatory response, during infection. So when immune system has to activate, At the same time, this protein can also activate. Now, just to understand how this protein work, we need a little bit more information. Otherwise, these are the proteins, and it help to prevent infectious disease. That's it. You only know there's a protein they create, but you don't know how it. So it is better sometimes to know a little bit the background, then you can better understand the concept, and even for questions, certain facts are missing if only we learn the very basic things. So certain areas we need a little bit to know about the theory for better understanding. Okay, I will write it like this. There is a protein, complement protein, and this protein is named as a C. C is a complement protein. And this protein has a number, it is called C3. Now you can understand there is a protein in the plasma, complement protein, its name is C3. Now the C3 is inactive. At the moment, right now, my plasma and your plasma, C3 protein are there as inactive form. Before we come to the how it can get activate, we can write like this. So such situation, it can activate. A is for activate by enzyme. So enzyme can change the structure of the proteins and it become active protein now. It's similar to blood clotting. So you can little bit recall your blood clotting mechanism, how it works. So complement protein exists in blood as a inactive form, and enzyme can activate, then it become activated form. Okay, now you're coming to the point, enzyme. So what will activate enzyme? If this enzyme activate, then it convert. So I will just briefly write, they are pathogen and damaged tissues.